Thank you very much for that wonderful introduction, and thank you, the organizers, for inviting me. I'm really happy to share our work. So probably half the people in this room do some kind of sequence to function modeling, but so I recognize not everybody works in regulatory genomics. So in regulatory genomics, what we mean by sequence to function is we would like to take DNA as input and output functional genomics tracks. So this would be histone modifications, the location of polymerase or the actual gene expression. And typically this will be some kind of very large neural network. And these models have gotten increasingly powerful over the years, culminating in recent advance of AlphaGenome, which outputs basically all of the functional genomic assays and outperforms all the other models basically on every task. All right, so this just happened. It's really exciting. Who's here already used it? I've been using it a lot. I think it's wonderful. So are we done? I would argue that maybe not quite done yet. So my group actually does quite a bit of work in evolution, and I take a lot of inspiration from that. And so I'd like to pose the question, what does actually true genome comprehension look like? I think this is quite nicely encapsulated in this very famous painting by Magritte, where an artist is looking at an egg and painting the bird that would eventually emerge from the egg. So maybe he can see the future, or maybe he just really understands biology. So we can ask a question, given just a DNA sequence, right? If you really understood the genome, you should be able to draw an elephant if that is the elephant's DNA sequence. All right, so obviously we can't do that. What can we do today? Well, today if you were given this task, you would just blast the sequence against like a standard database. And in this hypothetical example, that there's no elephant in the database, so your next best guess would be a manatee. So that is actually what you would draw. Clearly there's more to do, and this is a pretty contrived example, but there are some things that we actually do understand really well in this kind of evolutionary genotype-phenotype space. So one of my favorite examples is you may notice that you don't have a tail, but many of your mammal cousins do. And quite recently, there's been a lot of progress understanding exactly why that is. So the no-tail phenotype emerged in the ape lineage. And this paper really dissected the molecular mechanisms that give rise to this. And what their work points to is that there's an important role of this gene TBXT. And in the ape lineage, there's an allo element here, but along the evolutionary trajectory, there's a second allo insertion that is actually can form in the RNA, can form this loop structure. And this affects the splicing of the gene in such a way that you actually get exon 6 spliced out in about half the time. This paper quite nicely showed that both of the allo sequences are actually required for this to happen, and they were able to didn't give humans tails, but they did get rid of mouse tails with actually quite high penetrance by just introducing this. So this is the kind of thing that we want our models to uncover. So I'm very curious to see what AlphaGenome thinks about this locus. Here's the AlphaGenome output for this gene. And I really want to know what happens if I delete one of these allo sequences. This is going to show me a big change in splicing, but very quickly I realized this question actually didn't make any sense because already in the reference genome, it doesn't show any exon 6 exclusion, right? So actually it's like predicting the ancestral state for this locus. So this interpretation, this organism actually has a tail, but this is a human genome. So this is actually, I was not totally fair because TBXT is expressed in a very special context, and actually in most standard contexts, it's expressed, it's there, but it's quite low, so it's maybe not surprising that you're not able to learn really the fine mechanistic details of this locus. I mean, this is, of course, a totally solvable problem, right? We can just throw in a little more data that's actually kind of context specific, and probably with very little work, we can get the model to do accurate predictions in this locus. But this is just to highlight that I think there are more things to do. There's actually more mechanisms than there are genes. So we're not quite out of a job yet. And this, in parallel to all of these efforts for building bigger models that consume more data and have more parameters and output ever increasing amounts of different kinds of tracks, there's this kind of parallel momentum in the field where people are starting to build what we call interpretable models, which are small and output maybe just one track or just even like a parameter. And so really kind of on the opposite part of the spectrum, and lots of people have been working in this space, our group included. And so what can actually these interpretable models tell us? So one thing is interpretability bridges prediction in biology. And it's not as much about performance as really trying to get into some insights. And for these reasons, actually less predictive models can actually, in my opinion, can be really quite useful if they tell us why, or they tell us why they're less predictive. And in particular, even like failure to predict something accurately in an interpretable model can actually lead to some insights. And so for the first part of my talk, rather than just focusing solely on what my group has been doing, I'd like to highlight some recent progress by others in this field that has really shaped my understanding of kind of what interpretable models can and cannot deliver. And so I'll begin by talking about this kind of interesting prediction problem that I think come onto the stage relatively recently. This idea of predicting transcription start sites. So we have all of these assays that can measure transcription, the start of the transcription at base pair resolution. And maybe you haven't seen this before, and certainly I was a little bit surprised when I saw it first, is that transcription start site is not a single base pair position, like the one that you get of the GTF file, right? Actually, transcription starts in a broad region, and usually what we call the canonical TSS is the peak of this profile, but you actually get transcripts from all of these nearby regions. And what you observe in the data is actually a quite complex profile with ups and downs and little dips. And this is not a technical artifact. This is incredibly reproducible. And the observation that people made is that this profile is actually a treasure trove of mechanistic information. So, and it turns out, as it often happens in science, lots of people made this observation at basically exactly the same time. And so we actually have 3 transcription initiation models. So we have ClipNet and ProCapNet and the suite of Puffin models, which is actually a deep learning model, PuffinD, like a black box model. And Puffin, without the D, is the interpretable counterpart. And that's, the Puffin paper is really mostly about the interpretable model. So one thing to notice is all of the papers, they have a figure that shows you the model architecture. So for the first 2 models, I've highlighted the architectures here, and they take kind of a stereotypical shape where they show you kind of the basic building block of the architecture, and then it says ... and do this for 8 or 11 more layers. Now, in the case of Puffin, their architecture looks like this. There's no ... because it only has 3 layers, right? So it's not the most accurate model, and here's actually in the ClipNet paper, there's a comparison with Puffin. ClipNet is performing better than Puffin. But there's something satisfying about having this very nice model that you literally can look at the entire thing in one figure. So we can go a little bit in depth about what's going on here. So it starts out kind of in a way that basically any DNA sequence to function model starts out by identifying transcription factor motifs with small convolutions. So you can see here, this is a transcription factor activation layer. And all the different colors represent different transcription factors. There's actually not that many in here. There's just 10. And then, so this is kind of the part that is actually a departure from how we would normally do this. These transcription factor activities are turned into what they call motif effects, where now they apply a very wide convolution kernel that basically captures exactly how this particular transcription factor engages the polymerase. And then the final output is you sum all of this up and you exponentiate, and that gives you the predictive profile. And so in this main figure in their paper, this is an example of prediction and the experiment, and you can actually quite nicely get these very complex shapes in a relatively simple model. So we can look a little bit more in depth about what is learned in this model. So obviously, it started out with motifs. The motifs look like this, and they all have names, and they're all quite recognizable as very kind of like canonical, almost textbook transcription factors that are involved in transcription, like TATA and YY1. So the motif effects are here, are represented here, and they have kind of some complex profiles, but just a few observations about these. They're basically two directional motifs. So this is a TATA and YY1. And these are basically the motifs that activate transcription much more strongly in one direction than the other. So these are the ones that give you strand specificity. And then we have these more general transcription factors that their effects look really symmetric, so they don't care about strand. But the other interesting observation here is that transcription factors in this class, they're activating when they're upstream of that peak TSS position, but they're actually inhibitory when they're downstream. And so this was quite a satisfying observation. Just a few months after a Puffin paper came out, another paper came out from now experimental group. Basically, they came to very much the same conclusions about this paradoxical role for transcription factors depending on position. And I think it's really hard to overstate how much this is really an advance. Right? So for a long time, you would hear like, well, transcription factors can be activating, inhibiting, and it's context dependent. But somehow that's not really a satisfying answer. Like context dependent, what does it mean? Is it the cellular context, the context without the position of the other transcription factors? So all of this work collectively really nicely points to this very stereotypical position dependence. But of course, actually, it turns out we didn't really need our interpretable model to come to this conclusion. So, so this is the TATA parameters from Puffin, and the ProCapNet paper has a figure that's actually very, very similar, right? So we have the TATA motif displayed a little differently, but essentially the same information. And this is the effect on transcription initiation for TATA. Again, you will notice it looks strikingly similar. And indeed, the ProCapNet model actually has a lot more motifs. In fact, it has multiple different TATA-related motifs with their own kind of motif effects, so to say. And in some senses, like a richer model, so there's more opportunity to gain insights. Right, so so what is it like? What in my mind, there is actually still something that differentiates Puffin from these other models. And what's interesting about Puffin to me is that it doesn't just give you these kind of elements, it gives you the composition rules. It gives you extremely interpretable composition rules. So this is true story. I put this on a final exam for a class, a genomics class I was teaching. This was a question. They all saw the Puffin talk, and they also had access to all of the figures in the paper. And basically, I'm just asking, if you position these two motifs of 50 base pairs apart, which would give a stronger promoter, TATA followed by YY1 or YY1 followed by TATA? And so basically, everybody got this right. Turned out this was one of the easiest questions on the whole exam. And basically what this tells me, and in some sense, the Puffin model is human computable, which is like one of the strongest definitions of interpretability you could have. Just to kind of illustrate a little bit what's going on, here's, we have a static TATA site. In red, you can see the motif effect is actually offset. So the transcription is going to happen downstream. And we're sliding YY1 kind of through the sequence. You can see at one point, there's like a very sharp increase in TSS activity. And so we have like a very strong position-specific synergy in this model, even though the actual mathematics of the model is really quite simple. So I'm really struck by this because I think this is a really kind of nice and generalizable principle for sequence to function modeling. And you can get all sorts of interesting pairwise effects by just using this general framework. So for example, let's say I have two transcription factor motifs A and B, and they like to bind together with a very kind of strict grammar, right? So they want to be exactly either 5 or 11 base pairs apart, and everything else would be non-synergistic. So I can actually do this in this framework quite easily. So for one of the motifs, let's say motif A, I make the motif effect and I just offset it by 5 and 11. And now I will get a synergistic effect exactly with the right spacing. So just to point out a couple of things, it's not a unique solution. Obviously, I can switch those as transcription factors. I can do some other things as well. And also, this is a silly example because the motifs have width 1, which is obviously not realistic, but just for illustration purposes. Okay, so I think this is really powerful. We've been having a lot of fun in my group fitting these types of models. It's it's fun to see where you can get to the work and which things have hard grammar versus soft grammar. So this is a really exciting part of Puffin, but there's more to it actually. So there's a part of Puffin that's a little more confusing, right? Other than these transcription factors and effects, which, while we don't really understand the biochemistry of them, I think like you can start making some plausibility arguments. Transcription factors, they recruit the polymerase, but if they're like downstream, maybe they interfere with the processivity of the polymerase, something like that, right? Not a biochemist, presumably some of you have better answers. So that is all at least like very logical. But Puffin also contains this trinucleotide effect layer. And basically, this is a layer that is just an exhaustive list of all the possible trinucleotides. And they also have effects. And in this plot in the paper, they show that this actually in aggregate contributes quite a lot to the total prediction for transcription site activity. And the other thing to notice is that most of the trinucleotides that have strong effect contain a CpG dinucleotide within it. All right, so this is Puffin. That's maybe a little bit old news. And then more recently, we have new developments in promoter modeling. So the promoter AI paper came out just maybe a couple months ago. And so this is a new model for promoter sequence, that one that's really focused on variant effect interpretation. And one of the really nice things about this paper is they've compiled a large list of benchmarks that are publicly available. So if you have a promoter model, you can very easily test them against all the promoter AI benchmarks. So in their main figure, they highlight 2 benchmarks. Which are quite different. So one is the GTEC outlier benchmark, and this is rare variants, rare promoter variants in GTEC that have large effects, and the authors did quite a nice job actually at discovering these. And so these are natural variants, so we might expect they're quite depleted for anything that's really strongly deleterious or anything that's really like affecting a core promoter element. And then we also have the saturation benchmark, which, as it says, is saturation. So actually all variants are represented, including ones that obliterate a major core component of the promoter. Okay, so where's Puffin on here? Well, it is here. So this is Puffin D. This is the deep learning version of Puffin. It's not doing particularly well. The Puffin, the interpretive model, is actually not represented. And, I was really curious to know, well, why is it not there? Is it because it's doing really well? Or is it because it's doing so poorly that nobody even bothered to plot it, right? And I like to kind of preface everything I had to say. The only thing I had to do with the next few slides is just asking John some questions. So this is all work that was done by John, the senior author of Puffin. Okay, so here I just try to kind of reproduce the color scheme from the original paper as closely as I could. These are their tables. We're going to add Puffin. What do you think? You ready for the reveal? Is it really bad or really good? Okay, it's not good. This is Puffin. It's not good. Okay, so it's actually doing quite bad on low variant, on the rare variants. And actually this makes a lot of sense because like I said, the rare variants rarely hit these core promoter elements, which Puffin is composed of. It's actually doing reasonably okay on saturation and outperforming the deep learning counterpart. So, okay, it was maybe kind of a mixed bag of results. Okay, but then John was like, I fixed it, or, I didn't fix it. I made it a little better. And this all happened within the span of like 2 minutes. So he definitely didn't do very much. He didn't retrain anything for sure. It was just like, I just modified the model real quick. It's now doing a little better. I'll give you a second to try to think what he did. Okay. What he did was got rid of the trinucleotides. Right, because it's like a separate layer in the model is perfectly factorized. You could just like not consider that part of the model when evaluating variant effects. And this really produced an across-the-board improvement in all the different benchmarks. And some of them it was like here was quite a bit. Some of them was more than others, but it was actually incredibly consistent improvement across all of the benchmarks. So what is going on here with these trinucleotides? Okay, I think it's actually a really complex question. It's going back millions of years of evolution, but I'll give you my explanation. And if you have a better one, please let me know. I think there's probably more than one thing here that's correct. Methylated ECGs, like most CPGs in mammalian genome, are going to be methylated. Methylated CPGs mutate very easily via deamination, so they tend to kind of, over evolutionary time, they tend to go away because they mutate. But there are special regions in the genome that are unmethylated, and unmethylated CPGs are protected from this mutation process, so they mutate less readily. And lack of methylation also very nicely corresponds to functional regions in the genome. So just to illustrate, it's actually hard to find figures that plot all of this together. So this is the one we made for review, just to illustrate this. So on the top here, we have a methylation track. So you see most of the time methylation is high, and you see once in a while it dips. The bottom purple, this is just CPG density on the genome. Hopefully you can appreciate that things dip when there is a high CPG density. Not making any statements about causality, just association here. And also, if you look at a couple of functional tracks for enhancer and promoter marks, you can see very nicely that these regions correlate with a promoter or an enhancer. So what does this all mean from the perspective of a sequence to function model? Sequence to function models are very happy to learn that CPG regions predict regulatory activity. That is just statistically true in the genome. So these are very useful features, but they're not necessarily causal, right? We have this kind of evolutionary process confounder. And as a result, sequence to function models that are trained on actual genomes that have gone through the process of evolution actually are not 100% causal, right? And so this is actually really not an original idea. I think a lot of people have complained about this, and that's why many people think, in fact, we should train on synthetic sequences and MPR arrays. And but one thing I really like about this is like a really elegant demonstration of this this effect, in part because in the Puffin model, we were able to actually intervene on the model and improve the model by removing this confounder. And also, I think it's important to keep in mind, this is like a nice, clean example. I think we understand what's going on. There's probably a lot more things like this. So the fact that a sequence to function model is causal cannot be taken for granted. Okay, so just to conclude this first part, temporal models may not outperform black box models, but they do offer us some things. Shortcuts to biological insights. They also have reusable components that, either parameters or just kind of functional forms that we can apply in other contexts. And sometimes they can be manipulated without retraining to great effect. And so with this kind of background in place, I'd like to shift focus to a problem that our group has been working on, where you see a lot of the same themes emerge, but the context is quite a bit different. So the problem we've been working on is long-range regulation. So all of these promoter models, obviously like hyperlocal models, just looking at 1,000 base pairs around promoter, maybe a little more. But of course, in a mammalian genome, that is not enough sequence space to explain all of the specificity of expression and its timing, right? So a mammalian genome, gene expression kind of is often depicted like this in a textbook, where yes, there's a core promoter element, but there's lots of actually distal chromosomal elements that contain other regulatory sequences that loop around and come in contact with the transcription machinery that is assembling the promoter. And for about a decade now, we've actually been able to measure this 3D organization with quite high accuracies. So what I'm showing here is a kind of a standard contact map. So on the x-axis, you have kind of chromosomal coordinates in order here, symmetric matrix. And the color represents how likely those two loci are to come within a ligation radius. And so obviously, like if you're actually linked on the polymer, you're very likely to ligate. But we also see these regions that are actually far on the kind of the polymer string, but are actually still quite likely to ligate, like something like this. And we think this precisely represents this textbook picture where we're seeing like a promoter enhancer loop. So the genome-wide contact patterns, they don't seem random, but they have some structure. But the structure actually is not. It's not a low-rank structure. It has some complexity to it. And so the prevailing model currently for where the structure emerges is the loop extrusion model. So this model dictates that actually this genome structure that we observe on this megabase scale is orchestrated by a non-equilibrium process where a cohesin complex, you can think of it as having kind of two parts, like a left and a right leg. It binds on the chromosome. And what it can do is it can move its legs forward. And as it moves its legs forward, it extrudes a loop that gets bigger and bigger. The rate at which it can perform this extrusion is not constant. It is actually affected by various chromatin features. So one of the most kind of important chromatin features is CTCF binding sites. So they cause this process to stall. And quite fascinating, they do this in a directional manner, which is, this being depicted. We actually have two kinds of CTCF sites depending on their orientation. This one would stall progress in the right direction, but it actually would allow progress if there was a cohesin coming from this side. And this actually explains a lot of the features we see in the contact map. So when both legs are stalled, this creates a dot. When only one leg is stalled, this creates a line, right? So one location is stuck, but the other one is free to move around. And finally, as with all biochemistry, cohesin doesn't persist on the chromatin forever. At some point, it will dissociate and the process can start again. So you can actually make 3D models of this process and they recapitulate data really nicely. But it turns out you can also make 1D models of this process. And so this is a model that was proposed by Jeff Fudenberg, who's been working with us really closely. So you can just kind of simulate the cohesin without instantiating any kind of like 3D molecules. You can simulate how one cohesin contributes to a contact map. And so the way you do this is you initialize a cohesin somewhere with a loop of length 1. So this would be on the diagonal. And in any step, the cohesin can either do nothing, it can move one of its legs forward, or it can detach. And so depending on where it is and how long it resides there, you can actually start filling out this contact map, because as you move away from the diagonal, that just corresponds to bigger and bigger loops. Right? And so for this example, we put some CTCF sites there that are going to slow the cohesin down. You can see this one is getting stuck over here. One of the legs is getting stuck. We can get two cohesins going on at the same time, and you can see they actually got stuck at two of the different CTCF sites. So this is, of course, completely random simulation. And if you do this like 1,000 times, you can fill out a contact map that looks like this. This is starting to look like actual contact maps a little bit, but you could also venture to guess is that all that's going on here is that I put two different CTCF sites here, and I probably don't need to run 1,000 or 10,000 of these stochastic simulations to get this picture. Like, this is a very structured picture. I could probably just draw it, right, just from the information about CTCF sites. And indeed, this is what we did. So we've created the differential loop extrusion model, which is basically the steady solution to that 1D simulation, where the contact map is just described as a loop extrusion process that is governed by the speed at which the two legs can proceed. So that we actually have two speed vectors for the left and the right leg. And I'll just very quickly show you how this works. So we can look at a contact map and kind of we're considering the square in the middle. So this represents a loop of particular lengths. Well, how could you have gotten there? Well, in the previous step, you have to have a loop of length n-1. So you could have gotten here from either of these two locations. The probability of you moving between these is controlled by these rate parameters. And then the outflow from the cell is also controlled by the rate parameters, but with an index offset by 1. So, so this gives you a predictive model. Okay, but there's one more thing, and maybe it's a little fudge parameter, it's not super important, but we'll have to come back to it. The last thing that can happen is you don't just proceed on to the next loop, the cohesion can actually detach, so we have to add a lambda here to the denominator, but maybe lambda is a little boring because actually it's basically a global parameter. This is not, we're not modeling it as locus or sequence specific, right? So it's just a scalar. All right. This is what fitting a DLM model looks like. So you have your data. I'll be showing a lot of these. Data upper triangular, the fit is lower triangular, and the fit is actually just described by exactly these two speed parameters. So given these two speed parameters, I run the model I described forward, and I generate this output. As you can see, it actually fits the data really, really well. And just to kind of, as an aside, and this will become important later, we actually have been looking at data two different ways. So we have raw data, which is not negative because it represents kind of ligation probability, and hopefully is related to cohesion concentration in some monotone way. And the reason we typically don't look at data at the scale is because it really tends to hide all of this like little nitty-gritty details of where the loops are forming. And so, because most of the variance is actually explained by this like strong diagonal component, right, where the contact probability just decays rapidly as you move away from the diagonal. So most of the time we look at contact data in this observed versus expected form, where sort of the diagonal decay component has been normalized away, and this really highlights the fine scale structure. But of course, there is a con to this, which now this is a unitless quantity, so this is actually a departure from physical and biological reality. Like biology should have units. So, and we'll come back to this. All right, so one thing to just note is that we can fit the data on this kind of relative scale, and we can also actually account for the absolute scale. This is actually a little bit tricky, and you have to think about sort of terms that come from polymer diffusion, but not too difficult in the end. So overall, mostly I'll just be looking at the relative scale, just for clarity of visualization. This model fits the data really, really well. So what I'm showing here is we have 3 different loci in the genome. You see they look quite different with diverse patterns in them, and all of these diverse patterns can be fit with a loop extrusion model. We also have 2 different cell lines, so this H1 and HFF, and it's the same locus. And hopefully what you can observe is that I think you can tell that you're in the same locus. So there's some shared structure. There's also some cell type specific structure, and the model is able to fit the cell type specific structure. Now here's just visualizing the actual differences across the cell lines. And this actually works pretty well genome-wide. So this is the fits genome-wide. So for both cell types, we can kind of flip the cell type, so we can evaluate the fit across, with the wrong cell type, and you get something like this, where actually you still have some predictive power, right? Because there is some shared genetic structure, but the predictions are quite a bit lower, showing that we're capturing cell type specificity. And finally, this is just showing that we can capture the differences really well in terms of correlation. One fun thing about this model I won't talk too long about is that actually these rate parameters are scale invariant. So you can visualize this data kind of at different bin sizes and fit the model, and the parameters overlap really nicely. So once we have this model, we can ask the question, all right, so we have this model where we can reproduce the data in terms of cohesion velocity, but what actually controls it? And okay, well, one thing we already know is CTCF. So we actually built a multivariate model to predict the rate parameters. And indeed, our CTCF derived features are the, get the top coefficients in this model. And in particular, what we can observe is we make our CTCF features directional based on the motif orientation, because we know this is important. And indeed, the model is very nicely factorized according to direction, where CTCF plus is highly predictive for the left parameter and CTCF minus is highly predictive for the right parameter. And this is very consistent across the three cell lines I'm showing here. Okay, but what about, what else is there, right? CTCF, we knew that. So we can look at what else is going on after we subtract CTCF. And we see some kind of standard epigenetic tracks pop up. And the two things I'd like to draw attention to is we see a very strong signature for the promoter, actually, because it's K4ME3, but then a negative coefficient for K4ME1. So it's really trying to enrich for promoters. It really doesn't care about enhancers at all. And we also have a really nice signal for a mark that covers gene bodies. And so one thing you would predict from this is, if you were to get rid of CTCF, which is like a major cohesin block, well, what's the second cohesin block? Well, it's probably a polymerase. And so, right, it's another big motor protein on the chromosome. These things are going to interfere. And so one thing we would expect is for the cohesin to actually aggregate at promoters in the absence of CTCF. Thankfully, this experiment has been done. This is exactly what you find. So in this paper, they actually knocked out CTCF. So wild type, you can see these are cohesin components. They associate with CTCF sites. If you knock out CTCF, they tend to associate with promoters of genes. But our model actually made this prediction without ever having observed a CTCF deletion. So one thing about the DLM model is actually not a predictive model. This is actually just a compression algorithm. So if you give it some sort of new locus, it has absolutely no way of telling you anything about the locus until you show it the contact map. But we have extended this into a predictive context. And the way we do this is we have a model that takes in as input sequence features. And this is a context condition model. So it needs to know something about your context. And we use open chromatin as the context. And this allows us to do cell line transfer for the model. And so taking these inputs, we pass it through a small neural network to predict the left and right parameters. And from the left and right parameters, you generate your contact map. And this part of the model is completely fixed through the mathematical formula that I've already shown you. And like I said, this is a cell type condition model, so you can actually make predictions for a cell type you haven't seen before. And here we can compare our attempt for predicting an unseen cell type with C origami, which is another model that was made to do that. And you can see DLM here on the x-axis, and C origami are actually quite competitive. DLM doing a little bit better. And you can visualize some of the patches in the different performance regimes. So for example, orange is both models are doing about the same. And in green, we have DLM getting higher correlations. And red, we have C origami getting higher correlations. Now, moving on to kind of, I would say, maybe a more difficult problem, which is predicting a completely new region. And here we compare with ORCA, which up until AlphaGeno was state of the art of this problem. And so we don't outperform ORCA. I have to say the student was very upset. I was quite satisfied. I think we have a tiny model, only has 536 parameters. So I think we're doing really well. And like one interesting observation is that genome patches, there's a lot of genome patches that are just hard for both models. So there we see a lot of correlation in the performance here. Okay, so now we come to the final test of interpretable models. Can we actually modify the model globally? Could we make a prediction about a global perturbation? Right? This is kind of one of the promises of interpretable models. We should be able to do that. And so loop extrusion has been studied for a long time. It's a fascinating thing. People are very curious about kind of what are all the different components, how does it get perturbed under different circumstances. And so one of the important proteins that have been a focus of a number of different studies is this protein called WAPL. So WAPL is it facilitates the unloading of cohesin from the chromosome. Remember, like this is how the trajectory is going to end. At some point, the cohesin is just going to completely fall off, and so WAPL controls the rate of that process. So it's also a really highly constrained gene. So there's a PLI of 1, which is important to remember. And so the way these experiments are structured is the experiments are usually acute depletion experiments where you attach a degradation tag to the protein that you want to deplete. And this is really important because actually knockouts tend to have all sorts of secondary effects and actually can really confound the interpretation. All right, so here's some data from WAPL acute depletion. And so this is untreated cells, contact map. This is WAPL deleted. And I would like you to look at that and tell me if you can spot the difference between these two things. Anybody think it's hard to find the difference? Okay, the difference is there, and I think, you know, it's a quantitative, it's definitely not a qualitative difference. Like, it's not like everything just went haywire. No, we have a very subtle quantitative difference. We actually know that whatever WAPL does is really, really important because it's a highly constrained gene. So this quantitative difference in how is important, but is indeed very subtle. But we set out to answer a question, well, can we recapitulate this in our model? So, right, so what does our model say about this? So we know WAPL is actually going to control this last step, not any of these, like, you know, how fast you actually extrude the loop, but this very last step where cohesin unloads and detaches from the chromosome entirely. And in our model, this is governed by this actually lambda parameter, which is constant. So our prediction for deleting WAPL is that this should go down. And so we can test this directly. So first, let's look at the situation where we have, so this is our delta WAPL data, but the model was fit on untreated data, right? So, you know, these look kind of similar, but remember untreated and delta WAPL also look kind of similar. So they are different, and I like to point out why. It's a little subtle. So one thing is there's not enough mass as you move away from the diagonal in the untreated case, right? So there's kind of like, this is not orange enough. That's one problem we're having. And the other problem is if you look at these flares, the flares kind of don't go, I've cut them out here. The flares don't go far enough. They're not as prominent. Okay, so now what we're going to do is we're going to still keep our model that's fit to untreated data. So we've never seen this WAPL deletion, and we're just going to change lambda. And we get a plot that looks like this. So just by changing this one parameter, we've recovered the mass that we were missing, and we've also managed to extend this flare. And just as a sanity check, we can just fit directly to the WAPL depletion data, and it looks pretty much the same. But now this is like a whole new model fit with all new parameters. So, and of course, we can look at this genome-wide, right? So we have fit to untreated compared to delta WAPL, now showing actual error, because on the absolute scale, the correlations are always 0.9 no matter what you do. So here you really have to look at error. And here as a kind of, this is our transfer case, right? Where we fit on untreated data and just change this one parameter, and we definitely improve the fit. And this is a direct fit on the real data. And what I like to point out is there's actually no difference between these two in terms of predictive capability. So we were able to take this model and without fitting it to new data, modify it in such a way that it really reproduced this new context. So All right. In conclusion, I'd like to kind of draw some contrast between interpretal models and black box models. So black box models, they achieve really high accuracy. And I'm not sure interpretal models can compete with them. Definitely we can't compete with them for now. Maybe we will never be able to compete. And actually, I'm okay with that because we're just doing it for other reasons. So the things that we expect are just quicker insights and kind of these kind of reusable components or reusable parameters or reusable functional forms that we can plug into another task. Interpretive models have this interesting ability that, at least in some cases, you can adapt them to a new task not by retraining the model or fine-tuning the model, but by literally manipulating the parameters, sometimes in extremely simple ways, like just deleting them or doubling a parameter and getting the correct result. And one thing that I'm actually particularly excited about, and I think we're going to see a lot of this in the future, kind of hybrid models, right? And a hybrid model that I imagine is maybe a black box model. There's some black box computation that then predicts some interpretable parameter, like the rate of something or the concentration of something. And so it kind of retains the capabilities of both worlds. So and finally, I just kind of like to point out some challenges, right? We don't see so many interpretive models. In contrast, we have lots of black box models, relatively fewer interpretive models. And the reason for that is that it's hard. It's hard to make good ones. So in the black box kind of deep learning world, those models have lots of parameters. And what this gives us is there's actually many paths to an equally good solution, right? So the optimization landscape has all of these minima, and they're all basically equally good. They perform equally well on the test set. And that is the magic of deep learning. This is why it works. So we can do kind of off-the-shelf architectures and optimizations, and they just work. So interpretive models obviously have a few parameters, but they actually have a bigger problem. That for a model to be truly interpretable, it better give you the same answer every single time. Like, no matter which, like, half of the chromosomes you use, no matter how you initialize your parameters, it better have exactly the same answer. Otherwise, it's not the truth, right? So when you're working in this space, what turns out is that optimization really matters, right? You have to start now thinking about kind of, optimizing things in steps, optimizing some parameters while holding others fixed, and various other optimization tricks that really we were hoping like didn't matter anymore. So, and just finally, I'd like to thank all of the wonderful students and postdocs and collaborators that have contributed to this. So the differential loop extrusion work has been led by a wonderful postdoc in my group, Tina Šubić, and she comes from kind of a physics simulation world, and I think a lot of that intuition really comes through, and a lot of the work that she's done and the kind of these elements she pursued. And she was really adamant that we have to get the absolute scale perfect. I mean, I said, I was like, I don't care, but I was like, no, it has to be exactly right. I mostly skipped that part, but let me know if you want to know more about that. All of the deep learning or the predictive modeling was done by Turul Balci, who's now a postdoc with Sarah Mustafavi's group. I also like to thank a new graduate student, Kristina Perivoshchikova, and other members of the group. I'd also very much like to thank collaborators. So John Zhou, obviously, the senior author of Puffin. He did all of the extra Puffin evaluations that I showed you. Dennis Koska I worked very closely with in my department, and Jeff Gutenberg for lots of insights about chromatin folding. And thank you for listening.
Thanks, Maria. So we have time for questions. May you please dim a bit the light so we can see the audience? Or increase it? Yes, number one.
Thank you for a great talk and great work. The model tweak for the contact map was very impressive, and I wondered whether you can have a similar adjustment to test your evolutionary hypothesis regarding the CPG disappearance. Is that something that you considered or is possible?
So you mean in our sequence to function model? Actually, so our sequence to function model is really simple. It considers only CTCFs. So it's already extremely minimal. There's not like a CPG layer that I can take out. So yeah, the one I showed you is already super simple.
Thanks.
Hello, thanks for the great talk. I have actually a question about the pathing and the layers in the CPG.
Okay.
So CPG denucleotides per se can also be acting as a initiator. So have you observed like how did this interfere like with the appearance of the CPG in general, like over?
I think it's. I don't think this problem is solved, and I'm not actually claiming that CPGs are completely non-functional. I think I was very careful to never say that, and I don't think that is true. And I think there's a big kind of chicken and egg problem with CPGs in evolution because a lot of TFs actually have CPGs in their recognition sites. So this is a kind of like a difficult question to answer with there's no really true causality where everything kind of conspires to just work. I think the point I was trying to make is that there is definitely a confounding effect for CPGs, and you can remove it, and removing it actually can improve a model. And so it's just something we should be aware of. But I think, yes, certainly CPGs can also be functional.
So can just another sub question? How about did you maybe notice like a different predicting capabilities in the CPG islands versus non-CPG island promoters?
Yeah, so honestly, I didn't even do this. I was just right. So that is interesting to look at. We have not looked at that.
Okay, thank you.
Number 4.
Thanks for the amazing talk. So my question is, when we design these mechanistic models, and usually they are benchmarked on like some tasks and compare against deep learning models.
Mm-hmm.
But as you mentioned, the goal of the mechanistic models are not the best predictive performances. So should we build like specialized benchmarks for benchmarking mechanistic models? And how can we do that?
So I think to me, the kind of the ultimate test for a mechanistic model is to be able to do global perturbation predictions. So yeah, we don't have a ton of really standardized benchmarks for that, but there are some canonical ones that we can certainly reuse. For example, right, like for transcription initiation, you can delete some of those factors and observe that the profiles are different, and you can make actually quite reasonable predictions for how the profiles are changed. But yeah, I think for interpretable models, the test is global perturbation probably has to be tailored to a specific task, and hopefully eventually we'll have kind of like an arsenal of these that we can reuse.
Thank you.
I have a request to you guys. So my Juno seems to not respond. So if there are questions online, let me know. In the meantime, so I have a question for you. Mm-hmm. So when you approach with a mechanistic model, it's always based on some mechanistic assumptions. And perhaps also in the case of chromatin interaction, not everything is driven by cohesin, for example. Mm-hmm. So when you look at the differences, either between the observations and your predictions, or between the predictions with black box models and your predictions, where those are right, mm-hmm. do you identify different kind of mechanisms?
Right. Now, and not everything can be explained by cohesin, actually. So I think, right, so there's this, there's also just polymer dynamics that explain a lot of the data, and we're modeling it extremely naively, just totally homogeneous. All it gives us is like a distance decay, but actually, obviously, the polymer, there's phase separation and right, AB compartments. So there's we're kind of on purpose not modeling that. We like to leave the model factorized, but a lot of places where we make mistakes and like Orca gets it right are places where like the homogeneous polymer interactions matter. And,
okay.
Yeah, we're in the process of adding that as kind of a separate layer, but still keeping it factorized.
Thanks. Any other question? Do you guys see any question online? Sorry? There's this Juno, and there's a chat there and a Q&A. It just doesn't respond for me now. Okay. Well, thank you, Maria, again,
Okay.
for the great talk.